So today today is a soft deadline for your topic registration, and the topic that we're talking about is your grant proposal. So to remind you, you have to write up a grant today, grant, try to get money, do research, practice our writing skills again, communicating. That topic can be pretty much anything in biology or chemistry, as long as it has a molecular component to it. So if it's just chemical synthesis, that's not enough. You might have to say, well, we want to synthesize something and then apply it to pathways or something like that. I don't really care. If you are working on a research project now, go ahead and use that topic if it fits in molecular biology, as long as you haven't pre-written for grant. So this is new material you can't use an assignment you've already turned in for any other class for this assignment. I want you to actually practice writing. Okay, if you already got it done, and you're not going to get any more practice. Okay. Reason why I say it's a soft deadline is because I want you to think about what you want to write about. I want you to go look. If you don't know what research has been done on it, you better hit Google Scholar or something tonight. Look and see what's done. That's actually a good way to approach it. You can see whatever the latest research is, and then just say, hey, look, John Smith et al., they did all this stuff. Now here's what I want to do to extend that research. And that's typically what a lot of people do. They say, oh, I'm going to write this grant to expand or extend or add something new to the research. If it's totally novel, you as an undergrad will probably not get funded. So you want to stay away from stuff that's untested. We'll just save that for the Nobel laureates and all that stuff. Okay. So I'm not saying you're not capable, I'm saying it's hard to get funded. So with that, questions? Yeah.
So are we just sitting in the topic, or are we like writing out a whole thing?
Oh, yeah, right. One sentence to tell me kind of what you're thinking of writing on. And it's not you can change that. What we're trying to do is baby steps. So right now I want you to pick a topic, try to start getting papers for it, because if you look at the next assignment in the assignments list, it's the bibliography. So that is where you need to find 7 to 12 different papers on your research that you're going to use to write a grant. If you don't know what a grant looks like, I have examples, top sections for each of those. So you can go into Canvas, look at the examples that I wrote. I wrote an NIH grant, I don't know, a zillion years ago. So you can read those and see kind of what it looks like. I don't know if there's any online you can find as examples. But it's baby steps. So I want you to start thinking about the topic now. In a few weeks, that bibliography is going to be due, and that's going to be more of a solid deadline. So you don't want to put it off too long, because those other deadlines are pretty tight. So I want you to get it done, especially if I have to read 60 plus of them. That's going to take me a long time. So start thinking now. If you have an idea, great. Try to get your idea in this week at the latest, I would say. If you have to change it, that's fine. Just don't wait too long, because that'll leave you less time for the next part. And we'll eventually write the bibliography, the background. So the background section or the previous, usually what you start with is the story, right? What's already been done with your research. That's the background or preliminary studies section. And then we'll do the methods, and then that's pretty much it. We're not going to do anything else after that. Okay. Other questions?
This is more than the control of the bibliography. Let's say like we're doing this thing. Like you wrote up this other big sign, I think we've done the same thing. We're just papers that will help support science.
Yeah, there are papers that are going to help you write the grant. So they're either papers that are giving you background information on the system you're working in, or information on techniques you're going to use, something like that, or even the paper that you're extending, right? You can read the paper and say, well, this is the original, this is what we're going to do. But I think part of the bibliography is also putting your specific aims. Those are kind of your plans of action or avenues of research. It's not for me. The bibliography is not for me, right? It's for you to use. So I expect you're going to use most of those papers when you write the grant, when you start thinking of the grant. Because I'm not going to read the papers. 12 papers times 60? No, thank you. Anything else? this week. Hopefully you can get that in as soon as you can. So at least I know you're working on it. Start looking for those papers. If you can't find any papers under research, what do? Which topics? Yeah, which topics? Find a topic where you have background information to use. It'll make it harder if you have nothing to write on.
Where did you see the example? Right up for?
Sorry?
Where did you see the example right up for?
They should be in Canvas. When you go down to the assignments list, look at each assignment. I think I included, I don't know if I included an example of bibliography, but there's other sections of my grant that I put in. When you see something uploaded. I haven't looked at it for a while, but I'm pretty sure we'll have it. So assignments, I think it's way at the bottom. So if you look at preliminary studies, yeah, right there. Example preliminary studies. That's I think that's my NIH grant. Oh, well, I don't even want to see the date. It's so long ago. Okay, other questions? Okay, anything else? So I'm still reading through some of the replication papers, so I'll try to give you feedback this week on those, make sure you're on the right track, because we'll have another paper next week, or you'll have to do that again. Another summary for another paper. So if you're struggling with writing, let me know. Otherwise, I'll give you feedback if I need to. And what else? But that's kind of what the papers look like. You guys okay? Well, that's what we're going to do with each paper. So, so any groups that didn't participate last time, you'll be expected to participate in the upcoming papers, right? Everybody gets to go at least once or twice. I think probably twice, at least. Okay. So any questions on replication? Let's go back and review the major things we kind of talked about, huh? How do I get back to? I'm gonna get it back to the full screen. Oh, right over there. So, what was the paper about? So it's about initiation, right? So how would you describe initiation in replication? I wish. It's not simple, right? It's pretty complex, isn't it? Just with the few slides that I looked at, we are considering lots of different proteins involved. The paper alluded to this idea that these pre-initiation complexes have to manipulate nucleosomes. So there's multiple steps going on. Does it happen all the time? We just replicate and replicate and replicate and replicate. Hope you're a cancer cell, maybe. No, most of you are shaking your head no, right? So we have a lot of regulation, don't we? A lot of regulation telling us when to replicate. And then we have this idea of timing, right? Do they all go at the same time? And what was the major consequence with that? We have origins firing at different times. Faster, right? We got to keep track of them all, don't we? Because we can't have origins firing a second time while we're waiting for the other ones that haven't fired the first time. We don't want to over replicate. You don't want to under replicate. So there's all sorts of stuff, and I didn't talk about this last time, but there's a lot of research going on into this. Did you notice in the paper they talked about more than one origin? So there are origin sequences. So I'm going to list some things here that could affect timing. There's sequences of origins. They've actually sequenced origins, looked at some of them, and they found some sequences that are associated with timing. They actually timed them and saw that these origins might fire later in S space. Kind of interesting. So you could have a sequence effect. There's a weird paper I read that was on the what they call nuclear positioning. So we haven't showed you any pictures, but the chromosomes are not neatly lined up in the nucleus, are they? And we're talking S phase, so this is before prophase when they condense. You ever look up a picture of what the DNA actually looks like in a nucleus? It's just spread all over. So you could have some in the center of the nucleus and some on the periphery. Guess what? Origins that are kind of in the periphery, they tend to fire late. Tend to fire later. There's at least one paper that showed this. That's not proof, but it's interesting evidence that, wow, where the origin is in the nucleus could actually affect its timing. That's kind of weird, isn't it? To me, it is. It's kind of weird. Let's see, what was another one? Oh, I remember. We already talked about this. When do you think heterochromatin fires? You remember what I mean by firing, right? When they start bubble. Later, right? Why? What is hetero again?
It's more close, the nucleosomes are more closed up than I.
Yeah. So that's the tighter packed. Very good. Sorry, I didn't mean to put you off. It's tighter packed DNA tends to fire later. And that's kind of a good idea to know, right? Because those nucleosomes are interfering with the pre-replication initiation. The fork is there or something like that. Okay. So there's lots of cool things that could affect it, and there's still probably a lot of research that has to be done in this to really understand it. But what I want you to do here is take that step back and think about what are the consequences. You mentioned it. It's faster. Maybe there's a limit to the number of enzymes we have, so we can't fire all the origins at the same time. You know, all sorts of cool things you can think about that. But then ultimately, we have to make sure we don't over-replicate. Can't refire an origin that's already fired. Okay. What about elongation? I briefly looked at it. Very briefly, but I think I showed you these things here. Do we have to deal with anything in elongation? We might have damage, we might have rearrangements to deal with. There could be things during the elongation phase that the cell has to deal with. But what's the idea here? Can we just, I don't know, can we just give up halfway through if we have a bunch of issues and say, nah, forget it, I don't want to replicate. Okay, maybe some cells do that, right? And they go through apoptosis. But what's the ultimate goal again of replication? Make another copy faithfully, right? Faithful copy, high fidelity copy, as much as we can. So you want to think here that the cell wants to try to complete. So it tries to complete replication. No matter what. So even you can look up what's called the SOS response in bacteria, even in the face of huge levels of damage, the bacterial cell tries to finish and says, well, let's just try, because maybe, you know, it's less damaging for a bacterial cell to do that, no doubt. They can try to complete it, and if they have too many mutations, they might just die off. In our bodies, this kind of response might lead to cancer if we just try to complete it no matter what. But it kind of gives you an idea as to the goal for the cell is to try to make things work, to try to fix things if it can. And we'll come back to this when we talk about DNA repair. So we'll talk about some of the repair pathways that try to do this. Okay. Remember, a lot of this is food for thought. What about termination? And you don't have to memorize these different things. These are just giving you different ideas. And we'll look at some of these actual things, crossover events. Okay. What about termination in bacteria? Fairly easy, isn't it? Bacteria typically have a single circular chromosome. We typically have how many origins? Goes around the fork, or around the chromosome, bidirectional. And then it hits the terminated regions. So there's a lot of proteins called TUS proteins that will actually slow down the DNA and stop the forks before they hit. And then what's kind of cool You ever watch one of those magicians with the two rings? They have the two rings put together. They call them concatenators. Kind of interesting. So at the end, what we end up with here, I should get my little pointer, sorry. We have these two rings that are attached to each other. And so they actually have repair proteins that come in to cut the DNA, can't have them attached in bacteria, and separate them so that then they can go to the different daughter cells. Kind of cool. So they slow down the forks, stop them, detach the two chromosomes, and then you have to fill in these gaps because there's gaps on both proteins. So there's repair proteins to do that. Pretty simple in bacteria. What about eukaryotes? We have one single chromosome. No. We have linear chromosomes, right? Or at least most of us do. What kind of issues does that give us? So in termination, what do we want to say? I'll say prokaryotes, not bacteria. So in prokaryotes, you have to, what do we want to say? Finish replication and separate the two chromosomes. But what's the issue in eukaryotes? So bless you, you know that we require primers. There are RNA primers in the cell. A few of you put RNA primers for PCR on your PCR tutorial. Remember that those are actually DNA primers we use in PCR, so much easier to work with. They don't degrade on us, okay? So remember that. But if we have primers at the ends here, and we basically take those primers out, what are we left with? We're left with these big gaps at the end of the chromosomes called telomeres, right? At the end they're called telomeres. And that in a normal cell, this is what happens. So most of our cells, every time they divide, the chromosomes get shorter. The ends of the chromosomes kind of get shorter. And that might be okay for a little while, right? Until they hit a gene or something important that could lead to instability. But what's kind of cool is that we do have, not just right here in termination, for eukaryotes, the major deal we have to deal with is telomeres. So keep in mind, the status quo is that our chromosomes typically shorten over their lifetime. Every time they divide, our chromosomes shorten. How do we keep them from shortening? We use this protein called telomerase. Telomerase, discovered by Elizabeth Blackburn. She was at UC Berkeley. I think she won the Nobel Prize for this 7 years ago, probably 20 years ago now. What's the unfortunate thing about telomerase? Which kind of cells are they active in? Sorry, I couldn't hear you. I'm talking about what kind of cells, right? Cancer cells, which we don't like. Anything else? Do we have any normal cells? Untransformed cells, we call them. Some germ cells. Germ cells sometimes have telomerase, and one more. Well, maybe more, I don't know. One that we know for sure. Stem cells? You know what stem cell is? Stem cell. Okay. So wait, right? So that means most of our cells don't have telomerase active, right? Right, and that's why chromosomes shorten. What telomerase does, really interesting protein, is that it'll actually add primers on the end here and synthesize new ends of the telomeres to keep the chromosomes from extending. So here again, here's one of those fish experiments where you can actually see proteins attached or sequences of the telomeres. It's got the weirdest structure. Look at this. The DNA actually comes through here, curls around, and is inserted back into this little bubble. That's a typical telomere structure in a eukaryote. Of course, they differ slightly from organism to organism. And then look at the proteins. Oh man, including, well, if telomerase is there. look at all the other proteins that are found at the end of telomeres. Wow, I'd say that's pretty important, yeah? Pretty important part of the chromosome. Anybody have any hypotheses why we don't just let the ends of our chromosomes stick out like that? No, I'll give you the answer today, and we'll see the significance later on. Yeah, exonucleases, because what do they recognize? Ends, which are what? If it's just sticking out, it'd be a what? A double strand break, wouldn't it? It goes back to repair, actually. It looks like a double strand break. So that's one hypothesis. Why this is insert Vatican here is that those exonucleases, which are part of the repair systems, they don't recognize the chromosome as a break. Because double strand breaks, as we learned, are very, very bad. Okay. So just the weirdest thing, I forget, what do they call it? A T loop? Yeah. Called a T loop is what it is. Really weird. Okay. Let me just show you how telomerase works. You don't have to know the mechanism of this. But what I think is so cool is that telomerase, of course, is a protein, but there's another gene that codes for the template. So there's a gene that gives you an RNA template that the telomerase uses to do this coolest little mechanism. So two different genes that code for this, one for telomerase, one for this. That gene actually differs from species to species. So the ends of our chromosomes, the sequences all differ, but they're repeating regions. So what telomerase does, here's our newly replicated end with the gap. Telomerase will come in and it will use its RNA template to basically extend the 3' end of the DNA. Well, if we extend the DNA that way, what does that give us for polymerase? Or primase, actually, right? It gives us it's a 3' end, right? So we can build new DNA from 5 to 3 that way. And so we can come in and actually build new DNA on the end. And it's a repeating sequence. So I think this picture, do I have one more picture? Yeah, this picture shows it again here. So here's telomerase in yellow using the RNA template. It extends that end. This is on now the opposite side. And now primase comes in and you can make new DNA. Isn't that kind of cool? So you don't need to know the mechanism here. I just thought it was cool, so I have to show you. Pretty pictures, right? Okay. Yes, sir.
So I thought, and maybe I'm wrong, but like being replication behind this,
No. Primase always make DNA and RNA is always made 5 or 3, isn't it?
Yeah. Oh, like
So yeah, so primase doesn't have to start from a piece of DNA like polymerase does. Did I point that out last time? DNA polymerase 3 always has to start from a 3' end of like a primer. So primase doesn't have to do that. It's just able to put down single nucleotide, which is why people think it makes so many mistakes, because it can start from scratch. But it's okay, usually. Here we don't care, because there's usually no genes in the telomere. But with the other primers, remember, they are removed. So that's a good question. No, that's okay. And it's a good reminder, because sometimes we forget about little details like that. Like which way is DNA and RNA made? It's always 5.
And how is telomerase like regulated? So let's just keep infinitely going. To what extent does it stop?
Oh, wow. That's a good question. I'm not sure, but there are defined lengths usually of the telomere regions. I'd have to go back and see what that actual number is, though. That's a great question. Sorry, I don't know the answer to that. Cool. Okay. So expectations for this, of course, again, you want to go back and look at replication mechanism, so you're familiar with it. And then for our class, mainly, you want to take a step back and consider the whole process and the complexity we have to deal with and the regulation involved in these steps. That's kind of what I want you to do here. So especially something about, our initiation, when we looked at the paper, we talked about timing. Think about the things that have to go in at each of those steps. So rather than memorizing all the proteins that are involved, we're now just looking at the overall situation, okay? So that's kind of the idea for questions. I know, I'm sure I make your head hurt sometimes, right? With all the thought that I want you, all the things that I want you to think of. Okay, so any other questions on that? Again, you don't need to know the mechanism of telomerase. I did have a couple more slides to show. So one was there's one paper at least that looked at chromosome length and age. And I don't know how convincing it is, but they tried to put a little pretty line in there to try to show that as you get older, your telomeres get shorter. So there's at least some evidence to that. And that's linked to the theory of aging, right? If your chromosomes get too short, typically they become unstable, and so those cells die. So, but none of you are old, so that's good. You're all young and invincible, right? One of you can figure out how to make telomerase turn on in your bodies and keep you alive forever, right? Sounds like a horror movie, something. It always goes bad when we start, manipulating the genome in the movies.
So would that be why like people who spend a lot of time out in the sun, like if they're like a farmer or something, generally tend to like age quicker because their skin cells are being damaged more, they have to replicate more, and so then their telomeres are getting used up faster?
Yeah, I mean, this could be part of that. But then the other part is you just get a whole slew of mutations as well, right? If you're out getting a lot of UV damage. So I don't know which one's worse, but they're probably neither of them are probably good. Yeah.
Is the mechanism for aging based on telomere cell life? The telomeres are somehow regulating gene expression in the chromosomes themselves, or is it just like this telomere is shorter, so the cell dies faster?
No, they can. There are some interactions with these telomeric proteins. They do affect some expression of genes toward the ends, yes. But I think it's just the instability. If you get too short, you're running into coding regions at some point, aren't you? So you're going to damage genes. I think that's really the main point of this. And it's not the only thing, right? There's other things that damage our bodies. Anything fun, basically, right? Yeah.
I know along with that, you really documented cases of those genes being chopped out on the end and adverse effects because of that.
Oh, I'm sure. Yeah. Just hit Google Scholar or something. Yeah. He asked, are there any, I'm sure there's probably genetic diseases that are associated with it, I'm guessing. I don't know, but I'm sure there's documented cases of shortening chromosomes that show mutations. Yeah. I'd be very surprised if there isn't, but so I hope you can find some. Be wrong, I guess, but I'm sure there are. Yeah, sounds like a grant proposal. Look into telomeric regulation, and we can duplicate topics. With 60+ students, there's going to be some topics where people do the same thing, but I'm sure they're going to be different in some aspects. So I'm not too worried about that. And yeah, I guess I do have a review paper here. You can look up this review paper where they did look at some of the pathways that are involved with telomeres. And some of them do lead to cell apoptosis, which is death. And some cells actually go to senescence. So that's where they close down. They're not functioning anymore. So there are some links to disease, that kind of thing. Okay, so let's see. Should we do? I think I have a little reflection. I do this once in a while. So give me a reflection on this topic that we talked about in replication. So what things do we need to think about? What are some cool things that jumped out to you? You don't exactly have to answer this question, but I think it's good to think about what we talked about, why it's important, what's cool, what's new that you learned, that kind of thing. Then we're going to jump into the next section. Those little half sheets get passed around? Didn't pass them through. Did I come up short? Did everybody get one? Otherwise, you can find it on Canvas as well. I came up short, okay. That'll give you an idea of some of the enzymes that we're gonna look at in the next section, so. Yeah, regulation. Multiple origins, yeah. And that's actually some good questions, you know. If you only had one origin, so if you looked at 100 million base pair chromosome and you only had one origin at the end, would that make any difference with the amount of time it would take? If you're going maybe 100 nucleotides a second, it's actually a good kind of a thought question. You might want to go home when you think about timing or how long replication occurs. You know, why do we have 1,000 origins across the chromosome if it's such a pain in the butt to keep track of them all, right? Why don't we just have one? Or maybe even one in the middle. I'll give you, and you're right. The hint is it takes a long time. Doesn't really matter where you are on the chromosome if you just have one origin. I forget what the numbers are, but it's like years or something. Maybe not years, hours. Okay. Anything else? Any other questions on that before we get into the next section? No? Okay, cool. Oh, I got to wait before I exit out of this. Sorry. So everybody get a chance to answer, get your participation points. No smiles back. This is the problem with Mondays. Everybody's like, it's freeze. It's gonna be freezing all week, isn't it?
I hope so.
I'm ready for spring. I love spring. It's my favorite season. Summer's too hot, winter's too cold. I like the seasons, but spring and fall are like perfect weather usually. No? Okay. If you didn't get a chance, just email me if you're having issues. Should be able to review these as well. Okay, so next section is on recombinant DNA. So now we're kind of switching gears. So we've looked at DNA structure a little bit. We've considered 3D things. We talked about how to copy it, some of the considerations there. Now, as true molecular biologists, I want you to get a good taste of how to modify, how to manipulate DNA, because that's what all good evil scientists, all good evil scientists need to know how to manipulate, right? So on this list, I didn't give you a complete listing. I mean, there's probably 100 different enzymes you can use nowadays. Just go to a website like neb.com. This is helpful. New England Biozymes or something like that. They or biologicals, I don't know. They have a whole slew, probably thousands of proteins you can buy for whatever purpose you want. If you ever run into an issue where you can't find what an enzyme does, just hit their website and search for it. There's other websites, but that's where I order all of them. But really here, what I wanted you to get an idea of is just the length, the depth, the width of the molecular biology toolbox, all the things that we can do to manipulate DNA for whatever purpose we want to do. And we're going to do a little bit of this in the lab as well, okay? So the term here is cloning, right? So in molecular biology, the term clone does not mean like Dolly the sheep or anything like that. It's just basically manipulating DNA or changing DNA. And you don't always have to be cloning a gene. It's basically any kind of DNA that you want to work with, whether it's a promoter, a gene, or an RNA, whatever it is. But then you can use those pieces of DNA, you can use things like PCR to then manipulate or use those in expression studies or whatever kind of RNA interference, whatever kind of experiment you want to do, okay? So we're going to kind of go through this, give you some examples of some of the enzymes, especially on that little paper. Those are the ones I really want you to be familiar with. So probably want to go look them up, write down what they do, look and see. Sometimes there's good videos on what they're actually doing so you can see what the enzymes are doing. And I'll give you some examples of questions that I'm going to ask you as well, okay? So that's kind of what cloning means. Recombinant DNA just means changed, right? So almost like a recombinant chromosome is changed from its previous or original piece of DNA. That's kind of what the term means here. The main ones that I use are restriction enzymes. So these are enzymes that were originally taken out of bacteria. They got the name because they're part of the bacterial immune system. So bacteria use them to restrict the entry of DNA into the bacteria, foreign DNA, typically coming from viruses that are called bacteriophages. So bacteria use them to cut up just DNA they don't want. And of course, they have to differentiate between host DNA and foreign DNA. So it's kind of cool that bacteria can do that. But as good scientists, I didn't discover them. They were discovered back in the 60s and 70s. So we figured out how these things cut and how we can use them then to our advantage. And so that's what companies like NEB do. They find these new enzymes, figure out how they work, and then sell them for good money to scientists who want to use them. There's a couple of different types. The main ones we use or are going to use are type 2. So they're probably the most well-known. They cleave DNA. Typically, they cut double-stranded DNA. So if you have a piece of DNA like this, these guys are going to recognize a sequence, right? So there's going to be a particular DNA sequence that they're going to bind to. And the enzymes then usually bind like in what they call a dimer. So they're going to make double strand cuts on that DNA, depending, and I'll show you some examples. They don't always cut at the same spot, but they cut at the same sequence. So they cut both sides. It's usually a double strand break. Type 3 are a little weirder. They have more complicated restriction sites, what they call the sequences. Type 1s, I think, are the ones that just cut everywhere, so they're not very useful for our aspects. So we focus on type 2. They typically recognize 4, 6, or 8 bases, and that's kind of where we get these terms from. So if you ever see a paper that calls a restriction enzyme a 4-base cutter, that means they cut at 4 base sequences. So they cut a lot, probably, in a random DNA. So just when those sequences come up, okay? It's cool. They don't require ATP. So it's all through just the 3D action of the proteins. And they recognize a palindrome. I'll tell you what those are. And then they can have different cuts. So you can have a sticky cut or a blunt cut, depending on what the enzyme is. And so here's kind of what they're doing. The enzymes are cutting the phosphodiester bond. So they're separating the 2 strands on one side and the other. And ligase kind of goes the other way. So ligase is the one that will then actually can remake those phosphodiester bonds. So you can go back and forth. It's kind of cool. And you have to do that. You can't just use a DNA that's been cut, that's unstable at that point. You have to fix them, okay? Okay, what are palindromes? Right, so palindromes, if you look at this sequence here, they're going to read back, they're going to read the same way. So if you read it 5 to 3 on this one, they'll read the same way on 5 to 3 on the other strand as well. That's what a palindrome is. So type 2 restriction enzymes will recognize palindromes that are 4, 6, or 8 bases long, depending on the enzyme. And then you get some cool little examples here, right? Like, so never, odd, or even. If you read it backwards, it's never, odd, or even, right? So that's kind of cool. You can come up with palindromes that way, okay? And there's some funny ones too, but I'm not going to read them all. So here's an example of a protein. It recognizes CCC, GGD. If you look at the other side, what does it say? CCC, GGD, right? And then this particular one is going to cut right in the middle. So we're going to see that's a blunt end. Look at this one though, EcoR1 cuts at GAATTC, but they cut in between the G and the A. So that little arrow shows you where they're cutting in the phosphodiester bond. And we're going to see that this is going to create a different end from that kind, okay? Just tell me if I'm going too fast. Okay, so they bind palindromes. Where did they get their names from? Well, remember I told you that we extracted them from bacteria. So they're typically named after the bacteria and the strain, if there is one, and then the number. So if you look at like EcoR1, we got this from the E. coli strain R, whatever that is. And it was the first enzyme, the first restriction enzyme taken from that or discovered in that. So you can have an EcoR2, so I know there's at least 2 of them. Or HIND3 is the third enzyme from this particular strain, okay? So those are the names. And if you have to figure out what they cut, you just go to NEB. NEB, type in the 3 letters and the number, and then NEB will show you the restriction pattern. And why do they only show you one strand here? You can figure out the other strand, can't you? If it's a palindrome. Well, even if it isn't a palindrome, you know their strand is complementary, right? So again, remember the arrows show you where it's cut, okay? So you will get this on the exam. You don't have to memorize any palindromes, thank goodness. Okay, I'll give it to you. I don't even have them memorized, except maybe E. coli. But I will give you something like this, and you'll have to manipulate a little piece of DNA or something, as just like we're talking about here in class, okay? So you can look it up. They're given. We'll use some restriction enzymes in lab, so you get an idea of those, okay? How are we doing on time? I still got some time. 8 minutes. Okay, I'll go 10 minutes, all right? No, I'm just kidding. Okay, I'll give you some examples. I got another day for this, so we got more time. So here's HIM3. Where does it cut? So let's do this and make sure you can actually see what's going on. Sometimes it's good to write it out in practice, okay? So if the sequence is AAGCTT, TTCGAA. So this is 5 to 3, 3 to 5. And it says that it's cutting where? So it's cutting between the A's. So what I like to do is draw a little dotted line through here, so I can kind of see what's going to happen to the 2 ends. When we cut those 2 sides, realize what's the only thing holding this together? We cut the phosphodiester bond. Yeah, the hydrogen bonds, right? So those are pretty weak. So they can come off. Sometimes they can come back together spontaneously, but they don't stay, right? They're weaker than the covalent bonds in the backbone. So we just assume they're going to separate. So what does that leave on this side? We have a 5' and an A. And then the 3', what's on the bottom? CGA. So we have an overhang, don't we? We have a single-stranded DNA overhang, right? You guys see that? Sometimes it's confusing. So then the other side would look like this. You'd have an overhang on this side. And this is the 3'. So now, what kind of overhang is this? So let's do pair and share. Is this a 3? This is a sticky end. It's sticky because they both have single-stranded overhangs. But is it a 3' overhang or a 5' overhang? Convince your partner of why. Sorry, Jackson, I'm way over there. Okay. Sorry. I tried to go all. Just tell me if come on this side, share with this side. Okay, let me see what I have. Are there any extras on the paper?
This is your comment, right? I mean,
Okay, what is it? I know the answer's on the board. But if this is 5, then this end is 3, isn't it? Right here. This is 3, then what is this end? 5. So it's a 5' overhang, isn't it? And this one would be the same, isn't it? This is 3. 5, right? So think about the chemistry on the ends of these letters, okay? So this one would be called a 5' overhang here. What would a 3' overhang look like? KPN right here. So 5 and a 3. So again, it depends on where the enzyme cuts. So you want to pay attention to where those arrows are.
What are the implications of a 3' versus 5' overhang? Because 3' are how like free hydroxyl group.
Yeah. Well, can you religate a 3' to a 5'? So if I have, yeah, just between two pieces of DNA. So if I have, what is the other example? KPN is 5' GGTAC and 3' C. 3' C. Can I connect this to this? If my sequences were complementary.
Yes.
No, you can't. So even if I had the same sequence, so what would that be? TT, if I had a 5' TTCGA, would this fit with this right here? They would bump into the top, wouldn't they? So the implication here is if you're doing a cloning project, you typically have to match up 5' overhangs with 5' overhangs or 3' with 3'.
The recombinant DNA, we just like cutting, some sort of DNA, but cutting not a sort of DNA with the same nucleus.
Yeah. So a typical recombinant DNA or cloning project would be to cut out a gene of interest and then put it into a plasmid, for example. So we would use these kind of restriction enzymes to match that up, match it up and make sure the cloning process. Now, of course, there's ways to deal with this. So you can find on your list, there are polymerases or there's nucleases that you can treat these in with. We'll probably get to that maybe tomorrow, maybe next time, okay? But there are ways to manipulate this further. So we're just showing the beginnings right here. Okay, let's just do a few more. Any questions on that? I think this is confusing because I could give you this on an exam, right? And I could say, what do the ends of this look like? So you want to be able to do something like this on an exam or a quiz or something. Usually the exam's easier because you can draw on those. Okay, is that pretty straightforward? You might want to practice with a few, okay? What about a blunt end? So here, a SMA cuts in the same place. So basically, you just, I don't want to draw on this, just draw your dotted line straight down, right? And you get the blunt ends here. Can you still religate this? Because remember, ligation, so we could take this and treat it with ligase and put it back together, right? So ligase could do the opposite. What about with a blunt end? Do I have a question on that? Oh, that's a different question. Can you religate these? Yes, it's usually a little harder for ligase because without those little overhangs, then it's harder to get them back together, but it is possible. Usually you have to use more ligase and let it go for longer in the experiment. Okay, so we'll just leave you with this question. I don't know if we have enough time, but go home, take just a random piece of DNA. So this is a good way to practice. So here's a random piece of DNA and cut it with EcoR1 at one spot and BAMH1 at the next one. And we'll start with this question next time. Is that okay? So can you do that? Can you look up the sequences? You just take a random piece of DNA, cut them, and then see, can you take those two pieces and ligate the EcoR1 back to the BAMH1? And really what you're doing is you're deleting or cutting out that middle section. That's kind of what you're doing. Sorry, get out of the picture. And then start looking forward. Next time we'll look at some of these other enzymes, okay? How can we manipulate these things further? Okay. Quick question? Okay, thank you.